So one of the small intestine's most important jobs is absorption. I mentioned this in the previous video. The structure of the small intestine enables it to be good at absorbing. What can you think of some of these things? What enables it to be good at absorbing? I mean, literally it has lacteals, right? Absorption is going to be facilitated by having a high surface area. What are the ways in which the intestine is specialized with this high surface area? One is just length, right? 20 feet in a cadaver, when you've lost that tone, long, right? So high surface area. We've got time. You know, as you move chyme through, you're going to be able to absorb more if you've got a long tube to go through. Then we've got various structural modifications. First is circular folds. I'm going to go from like gross anatomy down to more microscopic. Here are the circular folds. You see them if you cut like a longitudinal section. We often look, you're in lab, you're going to be looking at a cross section, so you won't really see them. I can show you a picture. Maybe I should have done that to show you these folds. They are permanent, like the structures of the small intestine that kind of allow chyme to spiral through and slowing movement through the small intestine, so they're maximizing nutrient absorption. This is mostly absorption occurring in the jejunum. That's what I said before as well. It's consistent. Yay. Okay, circular folds. Then we've got our villi, of course, right? These right here, these little things, fingers, visible at like 100x. So these finger-like projections that are part of the mucosa, right? The mucosal layer. And then have that lacteal and the capillary bed in the middle. Lastly, what else do we have? Can you think of something? How about if I show you this? What is this? This is our microvilli, also called brush border. I think it's like painting a brush on the border. You still can't really see individual things here. We will look at these. If I zoom into this guy though, this is a cell, right? Let me zoom into the entire cell. This is a cell that came from like there. That's why we're zoomed in this view. If we zoom in further, we'd see like something like this. So you'll see pictures like this later. These are the microvilli. Those increase surface area. Can you see that? Microvilli are located on one cell. Villi are made up of multiple cells. One of the main functions of the small intestine is chemical digestion. Remember, that's the breaking of bonds, typically via enzymes, to break down stuff into smaller stuff that can be absorbed by the body, go into the bloodstream. Our goal is to make big things smaller. So what are these things that are chemically digested? And in this video, I'm going to emphasize the importance of the small intestine, liver, gallbladder, which is what we're focused on still here. So proteins, these are polypeptides made of amino acids. We need to break down proteins into amino acids that can be absorbed by the body. Carbohydrates. These can be small or big. So polysaccharides, glycogen, starch, long chains of sugar. Disaccharides means two, sucrose, lactose, and then monosaccharides, glucose. This is what we want to be able to absorb. And lastly, we will talk a little bit about lipids, so fats. Primarily triglycerides is what we take in, fats. There's also other things, so nucleic acids we actually break down. I'm going to focus on these three. I'm going to do these two in separate videos, like a walkthrough of digestion and absorption later, but we're going to do them briefly here. I will also talk about lipids more when we talk about the liver and gallbladder. But here I wanted to walk through these more broadly. So where does carbohydrate digestion, and one thing, I hope it's obvious, these are essential nutrients for our bodies to function. There's another chapter you can read about that talks more about that kind of nutrient-focused view. We're going to assume that we need these things. We need to make ATP out of glucose. There's actually pathways, indirect pathways of making ATP from these other components. We need to store energy as fat. We need to make new proteins with amino acids. So that's a whole other topic. We're going to assume we need nutrients. And we know that. So carbohydrate digestion begins in the mouth. There is something called salivary amylase that I've talked about before in saliva that breaks polysaccharides into disaccharides and trisaccharides. So it breaks down polysaccharides, that first step of digestion, chemical digestion of carbohydrates. If you chew a cracker, you'll notice it starts to taste sweeter and sweeter. There's extra credit related to this. And that's because it's going from like starch, I think is what's in saltines, primarily to disaccharides that actually taste sweeter, like sucrose. So carbohydrate digestion starts in the mouth. But most of it happens in small intestine. I'm going to put most in small intestine. Come back to that. Okay, proteins, you know this already. There's pepsin in the stomach that breaks down proteins, breaks down polypeptides. It is also going to then most in small intestine. Fats, there's actually a little bit that happens in the mouth as a lingual lipase, but I want to just go here, oops, wrong color, and tell you that most happens in the small intestine. So in the small intestine, what's happening? There are several sources. First, actually, I'm going to do these two, because these two require primarily pancreatic enzymes, as well as intestinal enzymes. They're produced by the absorptive enterocytes, but they're kept on the brush border. I'll show you these in a bit. They're called brush border enzymes because they live on the brush border of the enterocytes. Fats, primarily bile from where? The liver. Yep. So all of this is aided by secretions from the liver, pancreas, and the brush border enzymes. So that's where we're gonna dive into the liver and pancreas next. We are mostly talking about the duodenum, but some chemical digestion does occur beyond that as well.